Hello, this is Tim Seipel, Cropland Weed Extension Specialist at Montana State University. We're presenting a study that we did in 2018 and 2019 on cover crop trade-offs, where we evaluated soil moisture, weed suppression, and soil microbial communities in response to cover crops. The rotation began in 2012, and it's a wheat cover crop rotation. The treatment in the project was different mixtures of cover crops, and those different mixtures of cover crops had different phenologies. Either they grew earlier in the season or later in the season, and there was different diversity of mixtures. Here is what the cover crops looked like in the field in Havre. As you can see, there was different amounts of biomass, different structure of the species, and different timing of growth. We also compared the outcomes under ambient conditions with no modified weather or climate conditions. And then we also used these things called open top chambers and rainout shelters to block precipitation and make it warmer, so that we knew what conditions would be like if we encountered warmer and drier years. The cover crop mixtures we concentrated on were a 5-species early season mixture with an early phenology, meaning growing early, flowering early, and a 7-species mixture with a mid-season phenology, having more warm season species within it. In the table, you can see the list of the two cover crop mixtures for each year, and I'll talk more about that now. So the biomass of the cover crops in the early and mid-season were similar between ambient and warmer and drier conditions. But you'll notice the mid-season bars, the black and gray bar in the right there, is much lower than the early season mixture. The early season mixture produced much more biomass in a shorter period of time. The cover crops were planted in approximately the first week of May and harvested near the middle of July or the first to second week of July. Even though the biomass was similar, the relative abundance changed when we induced warmer and drier conditions. Here's the oat biomass, and compare the gray bar and the black bar in these two graphs on the right side. You can see in the warmer and drier condition, the gray bar, the amount of oat biomass decreased in both the early season cover crop mixture and in the mid-season cover crop mixture compared to the ambient conditions. But what you'll notice is the decrease in oat was compensated by an increase in the mustard species. So the amount of turnip there went up in the warmer and drier conditions, and so did the amount of radish. This highlights why it's good to sometimes consider mixtures of cover crop species, because you don't necessarily know how they will respond individually to environmental conditions. Here's an image of those. The left shows the oat growing under ambient conditions, and the right photo shows the increase in the mustards, the radish and turnip, under warmer and drier conditions. In general, the cover crops, there was more biomass within the early season phenology, so things that are C3 species that begin to grow early. Otana oat, Arvika pea, groundhog radish, purple top turnip, and hairy vetch produce the largest amount of biomass. In the 7-species mixture, we had 4 warm season species that were in there. They include Cheyenne soybean, sorghum Sudan grass, German millet, and frontier chickpea. But because of the timing when we planted these and the termination of the cover crops in July, they had very little time to accrue very much biomass and grow rapidly. When we look at the soil moisture graphs over the season, the bottom horizontal axis is the date, and the vertical axis shows how the amount of soil moisture is there measured in bars. As you can see in what we know, the fallow in the top left corner stays kind of flat. We're not losing soil moisture as rapidly, except for maybe under warmer and drier conditions. On the top right is the early season mixture of cover crops. You can see the rapid decline in soil moisture and then a slight increase that's out there. The mid-season cover crop mixture used less soil moisture because there was less biomass and it declined more slowly. And then in the bottom right corner is the winter wheat soil moisture. You can see it rapidly declines through the middle of the season, but then has a bump that goes back up. And that bump was related to the 2018 and 2019 field days at Havre, where very large thunderstorms with lots of rain came through in both years. So the early season cover crops use the most soil moisture, similar to wheat, maybe not quite as much as wheat, but similar, and the mid-season less and the fallow the least, highlighting the use of early season cover crop mixture. This did affect wheat yield, but not in all years. The left graph is the wheat yield in bushels per acre in 2018, and the right graph is in the wheat yield in bushels per acre in 2019. The black bar was the normal ambient conditions, and the right bar is the warmer and drier conditions. As you can see, that warmer and drier conditions reduce the yields from somewhere around 65 bushels an acre to about 40 bushels per acre in the fallow, early, and mid in the first year. You can also see that in the second year, 2019, on the right, that there was no yield loss in fallow to warmer and drier conditions, but in the early and mid-season, the 8 consecutive years of wheat cover crop rotation seem to reduce wheat yields considerably. This is what the warmer and drier conditions look like on wheat. You can see that there was definitely less yield. When we look at weed biomass was best suppressed in the early season cover crop mixture. So if you look at the graph on the left side, you can see fallow and the mid-season mixture under ambient conditions had the most weed biomass. But consistently, early season mixture did the best job at suppressing weed biomass, having much less weed biomass, the middle bars there. If you look at that carryover into winter wheat, the early season cover crop also had a legacy effect, and the plots that had the early season mixture also had the lowest amount of weed biomass. Here are arrows pointing to those, highlighting the ability of an early season cover crop to suppress weed biomass. We looked at soil microbial communities, and we looked at 2 types of soil microbial communities. We looked at the fungal communities, and we looked at bacterial communities in the soil. And we found clear shifts in response to cover crops in both bacteria and fungi. These graphics here are just used to demonstrate the similarity or dissimilarity. The less the circles overlap, the more different the microbial communities are. Specifically, In the early season cover crops, there were fewer plant pathogens and more beneficial fungi. On the left, we have the arbuscular mycorrhizal fungi, the beneficial fungi that connect to plant roots and help bring in nutrients and moisture. The richness of those, the number of species, was highest in the early season cover crop in the middle, lower in the mid, and lower in the fallow. And if you look over on the graphic on the right, that's the fungal pathogen abundance. And you can see that in the early and mid-season fungal cover crop mixtures, fungal pathogens were lower than in just a fallow field. There are trade-offs when we use cover crops, and I think we always know, but more soil moisture use and led to lower wheat yields. But the cover crops led to better weed suppression and more beneficial fungi and fungal pathogens. We can integrate cover crops into Montana, but we need to better keep in mind these trade-offs that we have to make. So I'd like to acknowledge all the people who have worked on this project, all the people at the Northern Ag Research Center in Havre, all the co-authors, graduate students, and the university, and then our funding sources, the Montana Wheat and Barley Committee and the USDA NIFA program. If you have any questions, feel free to contact me, or I'll also be available in the live session for the field days. Thank you.